Hello, my name is Mr. Chipman, and I am the AP Biology teacher at Murray High School in Murray, Kentucky. And today we're going to be looking at some common misconceptions in AP Biology, dealing specifically with gene regulation and gene expression. Hopefully you'll enjoy this. If you do, please give me a subscribe and a like down below. Hit the notification bell because I'm going to be making lots of videos as we lead up to the exam in May. So let's talk about gene regulation and expression. What is gene regulation and expression? Well, gene expression is when a protein is made. We've talked about that. We have DNA. DNA codes for proteins. How does it do that? Well, it's transcribed into an RNA, and that RNA is then translated into a polypeptide or a protein, which is a string of amino acids. So when genes are regulated, then that gene expression is controlled. There are various control mechanisms for gene expression, and those control mechanisms are basically going to say, we don't need this protein right now, whatever that protein is. It could be a developmental protein that's basically saying, we don't need the instructions on how to make 5 fingers anymore. Or it could be some sort of enzyme that the body or the organism doesn't need anymore, or doesn't need currently. Whatever the case, genes are being regulated because expression needs to be regulated. Let's look at some examples. So there is, first of all, in prokaryotes, the primary gene regulation mechanism is called an operon. An operon contains several different kinds of proteins that are going to regulate gene expression, and the most commonly used operons are called the lac operon and the trip operon, which is on the next slide. And let's just look at a couple of these examples. First of all, in the lac operon, it has to do with the presence of lactose. Lactose is a sugar that has 2 pieces, maltose and glucose. And if glucose is low in the cell and lactose is present, then the gene that is going to break down lactose begins being transcribed. How does it do that? Well, if there's no lactose, you can see that there's a repressor that is bound to the operator. The operator is kind of like the switch that says, let's do this thing. The repressor's stopping that from happening. And notice what's there waiting at the promoter. There's RNA polymerase. What's its job? To make an RNA, right? And so RNA polymerase is unable to move forward because that repressor's there. Well, you can see in the picture below, when lactose is present, lactose binds to that repressor, causes it to lift off the operator, and then the genes like Z, like Y, and like A are then able to be expressed so that lactose can be broken down. When that last piece of lactose is broken down, the repressor binds back to the operator. You get the idea. Trip operon is kind of the reverse of that. Tryptophan is an amino acid that needs to be synthesized in some bacteria because it's not present in their environment. Tryptophan doesn't always need to be synthesized. And so when tryptophan is present, tryptophan binds the repressor and then keeps it from being transcribed. But if tryptophan needs to be synthesized, obviously tryptophan is not going to be at the repressor. The repressor is going to lift off the operator. RNA polymerase is going to transcribe trip E, D, C, B, and A. How did I know that? Well, I just looked at the picture, right? I don't know what those genes are. I just know that they're involved in the synthesis of tryptophan. And so this brings up an important point. These are the 2 most commonly used operons, but you should be able to use this basic idea to transfer it and to apply it to any sort of operon. And so if you see something that you've never seen, don't get completely just flummoxed by it, right? Move through it, look for the things you know, understand what gene regulation is, understand expression, and then move through that, right? Kind of highlight the things that you know. Always an important thing. Now, one very important misconception is that operons are only prokaryotes. This sort of thing doesn't have anything to do with eukaryotic organisms, right? And so we're going to see some eukaryotic organism gene regulation, but prokaryotes are specifically, operons are specifically prokaryotic. So let's look at gene regulation in eukaryotes. There's going to be some differences. Eukaryotes have a nucleus. That nucleus is going to contain the DNA. The DNA is going to be bound up into this state called chromatin, and chromatin is going to be just kind of present in the normal life of the cell. And chromatin is essentially DNA that is bound up on these tiny little proteins called histones. histones are positive, DNA is negative, and they're attracted to one another. And if the gene needs to be expressed, then that DNA kind of unwraps itself from the histone and is transcribed, translated, the whole thing. Well, there are certain things that can kind of set on the DNA or set on the histone that is going to cause the expression or regulate the expression or repress the expression of a gene. And a lot of times you'll see this kind of all grouped together into this term called epigenetics. Epi means on, right? Genetics means genetics. And so it's like on the DNA. It is preventing or even advancing the transcription and translation of a particular gene. Epigenetics is a really wide topic. And so anytime you see a gene being affected from the outside, it could fit into this, right? It could fit into this category of epigenetics. Again, this is a big wide category here. I'll talk about it a little more in my videos for 6.5 and 6.6, and you can check those out. But just a wide category, much different than prokaryotes, which don't have chromatin, they don't have any of this sort of thing, and so there's nothing affecting genes from the outside. Now, transcription factors are another ways that eukaryotes regulate gene expression. And this has to do with the presence of regulatory switches or enhancers, is what you'll see sometimes here in this particular picture. You see that word enhancers. And the enhancers are going to, in the presence of particular environmental conditions, cellular conditions, any sort of thing like that, that enhancer is then going to promote or enhance the transcription of a particular gene. Why? Well, there's some sort of environmental condition that needs that or negates that. It could completely say, no, we don't need that switch turned on right now. And notice there's 3 different enhancers for this particular gene. Well, there could be 3 different situations in which that gene needs to be expressed, right? And so those enhancers are sections of DNA that are going to regulate the transcription, whether it turns it off, turns it on. And you can see all these other transcription factors, which is just kind of a catch-all for all these proteins that are involved in that, are going to bind to the promoter and then oversee kind of the transcription of gene A in this particular picture. How does this translate itself to real life? We'll talk about that in a second. But some common misconceptions here have to do with the difference between prokaryotes and eukaryotes, and I already highlighted that with operons already. But prokaryotes don't have a nucleus. And so transcription and translation oftentimes happen at the same time, as you can see with the picture there on the left. Whereas in eukaryotes, there are several things that are happening to that RNA before it actually makes its way out into the cytoplasm. That RNA processing, we talked about the 5' cap, we talked about the poly A tail, we talked about the introns and exons, and the introns being removed and the exons being combined. And so all of that has to take place in eukaryotes. None of that is taking place in prokaryotes. And so gene regulation in eukaryotes is going to be a lot more complicated process. Sometimes you'll have pre-transcriptional regulation, which we saw with epigenetics. You have some post-transcriptional things where there's tiny pieces of RNA, and I talk about those in my other videos as well. But anything that's controlling the expression of a gene is going to fit under this. Eukaryotes is going to be a lot more complex just because they have more things going on. Let's look at a real-world application for this. So here's a fish called a stickleback. If you're an AP Bio student, some of you have probably done the little activity that has to do with stickleback genes. Well, sticklebacks in marine situations have these spines on them. The spines have to do with this gene called PIDX1. When expressed, it causes the development of these spines in the pelvic region of the fish. Well, in freshwater systems where there are dragonfly larvae that like to eat little fish like this, those spines become a way for dragonfly larvae to kind of catch on to the fish, so to speak. And so in freshwater examples of this fish, those spines are not expressed. How does that work? Well, that enhancer that causes the PIDX gene to be expressed is absent in freshwater sticklebacks. And so this is an example of how expression and regulation are determined by the environment. And the fitness of this organism is going to be determined by the environment, and then gene expression is just going to match up with that. So as we move forward into Unit 7, you're going to see how a lot of these themes that we've talked about, particularly in Unit 5 and Unit 6, are going to really be fleshed out into the environment, and seeing how those variations lead to adaptation, which lead to increased fitness. Hopefully this video has helped you overcome some of your misconceptions with gene regulation. And again, I encourage you to watch my videos, 6.5, 6.6 in particular, dealing with those topics. And good luck on the exam in May.